Welcome to mitosis. What is mitosis? It's defined as the division of the nucleus into two nuclei with complete sets of chromosomes. However, mitosis is followed by the division of the cell and organelles as well, this resulting in two new cells each with complete sets of chromosomes. Function of mitosis. What are they? Growth of all tissues and organs after birth, replacement of cells that die, repair of damaged tissues. Phases of mitosis. The first stage is prophase. The second is metaphase, the third is anaphase, and the last is telophase. In prophase, the chromosomes appear condensed and the nuclear envelope is not apparent. In metaphase, thick coiled chromosomes are lined up in the center of the cell on the metaphase plate. Spindle fibers are attached to the chromosomes. Anaphase, the chromosomes have separated and are moving to altopoles. Telophase, the chromosomes are at the poles and are becoming more diffuse. Since it had passed all of these stages, it will come to a cleavage furrow called cytokinesis. Another stage is called interphase. Is broken down in three main stages: G1, S, and G2. G1 phase, the cell checks to make sure that the cell has grown enough to divide. If the cell passes the checkpoint, it will move into the S phase and start copying its DNA. If the cell is not able to move forward, it might enter a phase called G0. There is also a checkpoint at the end of the G2 phase. This is one last stand for the cell to make sure it has completely replicated its DNA. During mitosis, the DNA must line up in the middle of the cell so it can be distributed equally between the new cells. If the DNA isn't properly lined up, cell division will stall until the problem is fixed. If the cell cycle becomes unregulated, the result can be cancer, which demonstrates why this checkpoint is so. Proteins are also involved. Proteins that help start the cell cycle are called oncogenes. Proteins that stop the cell cycle, tumor suppressors. Remember how mitosis is formed by this diagram. Thanks you for listening.